Answering MCAT questions in under 60 seconds. Biochemists studying nucleotide synthesis observed that the N5-N10 methanotetrahydrofolate, a folate derivative, is essential for the enzymatic conversion of DUMP to DTMP by thymidylate synthase. This compound is directly involved in the transfer of a 1-carbon unit during the reaction, which best describes the role of this molecule in this process. Okay, so it sounds like this molecule, which is a derivative of folate, right, is necessary for this reaction to occur, right? And this is a 1-carbon transfer. So what is it actually doing? Okay, well, it acts as a transcription coactivator during S phase. Well, this is not talking about transcription at all. This is talking about some sort of metabolic process. So that's wrong, right? And specifically some sort of metabolic process where involving actual synthesis of a particular nucleotide, right? Whereas while nucleotides are of course involved in transcription, right? This is something that would have to happen before, okay? B, it serves as a competitive inhibitor of thymidylate synthase. Well, that's wrong because it's saying that it's basically facilitating this reaction that this thymidylate synthase is presumably catalyzing, okay? So it wouldn't be an inhibitor. And then C, it functions as a coenzyme in the catalytic process. Well, that sounds correct, right? Because a coenzyme is basically a molecule, right, that is helping the enzyme complete the reaction it synthesizes, right? It's not necessarily part of the enzyme structure, sometimes it is, right? But it is necessary for the enzyme to complete its function. So if it wasn't present, it couldn't happen. And there's a lot of different mechanisms that these coenzymes will participate in. In this case, it's like they're saying it's a transfer of a carbon unit, right? But if this molecule wasn't present, this enzyme thymidylate synthase would not work, okay? And then D, it binds allosterically to increase the enzyme's affinity for DUMP. Well, no, right? It's not saying that, right? That would be something like an allosteric activator. In this case, they're definitively describing some sort of coenzyme, right? An allosteric activator does nothing in terms of like participating in the reaction. It simply changes the structure of the enzyme in a way where it has higher affinity, okay? So that's wrong. So C is the best answer.